Okay, thank you so much for confirming. So, if you have any problem hearing my voice or seeing this screen, please write in the chat box. Okay, we will wait for 2 to 3 minutes, then we will start today's class. Okay, so very good evening to all the students who have joined today's class. My name is Moshumi Dev and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT in Dhanbad. This is the week 11 live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. Like every week, this week also we will solve some 10 to 15 questions related to the week 11 course content, and this kind of question will help you to understand the course better, and it will also help you to do your weekly assignments and also in your final exams. So we will just going to start in a few moments. So if you have any doubt during the session, please write in the chat box. At the end of the session, I will answer your queries. And also, if you have any problem hearing my voice or watching the video or the lecture slides, please also write in the chat box. Okay. And this lecture video and the presentation will be given to you by the NPTEL site at the end of today's session. So we will now start today's session, and if somebody wants to join, we will just let him or her enter. Okay. So the first question. So the for problem number one of today's session, it says that which of the following statements is false for the secondary metabolites? Option A, they are derived from primary metabolites. Option B, polyketides are one class of secondary metabolites. Option C, they are directly involved in growth, development and reproduction. And option D, produced in a small quantities. Okay, so among these four, you have to identify the false statement regarding the secondary metabolites. So the metabolites, which are of mainly two kinds, the primary metabolites and secondary metabolites. And in this question, you are asked for the secondary metabolites only. So we will just see the answer first, and then we will discuss about the secondary metabolites. So which one of the following statements is false for the secondary metabolites? The correct answer will be option number C, that they are directly involved in growth, development and reproduction. So this is the false statement regarding the secondary metabolites. Now let us see the secondary metabolites. They are the compounds which are derived from by the pathways from primary metabolic routes and are not essential to sustain the life of the cells. So these compounds do not have a continuous production, and very often secondary metabolites are produced during non-growth phase of the cells. secondary metabolites are the end product of many primary metabolites such as the alkaloid, phenolics, steroids, and polyketides, and essential oil, lignin, resin, tannin, etc. So these are the for the from this part we are sure that why the statement number C is false, because it is saying that it is directly involved in the growth, development, and reproduction. But actually, the secondary metabolites, they are not directly involved in the growth and development and reproduction of the cell. So this is the I have said before that there are many primarily there are two kind of metabolites. One is primary metabolite and other is secondary metabolites. So here is a table comparing these two metabolites. So primary metabolites, they perform physiological functions in the body like intrinsic functions, but the secondary metabolites, they are derivatives of the primary metabolites. Primary metabolites, they are directly involved in the growth, development, and reproduction, and the secondary metabolites, they play a ecological functions. The primary metabolites are formed during the growth phase due to the energy metabolism, and the secondary metabolites, they are formed near the stationary phase of the growth. The first one produced in large quantity and the extraction is easy, but the secondary metabolites, they are produced in small quantities, and because of that the extraction of the secondary metabolites are difficult. The primary metabolites are same in all plants, and in case of secondary metabolites, they are different in different plant species. A part of the basic molecular structure of an organism is the primary metabolite, but the secondary metabolites, they are not a part of the basic molecular structure of an organism. Ethanol, lactic acid, nucleotides, or vitamins are the examples of the primary metabolites, and pigments, antibiotics, drugs, these are the example of the secondary metabolites. Okay. So I would suggest that please remember few differences and few comparisons between the primary and secondary metabolites, because these are very important, and there are different classes of secondary metabolites which we will all look in our next course. There are pigments, antibiotics, drugs. They are very important class of secondary metabolites. Polyketides are also a very important class of the secondary metabolites. So I think this problem is clear, and then we will next move toward our next problem. So problem number 2, it says that the cofactor for the conversion of the acetyl CoA to malonyl CoA is option A, NADP, option B, biotin, option C, ADP, and option D, TPP. Now, what is a cofactor? We have discussed in our previous weeks, because what is a cofactor? So, which is a it helps basically helps the enzyme to do the work. Okay, is the intrinsic part without which the enzyme is not active at all. So, without the cofactor, the enzyme is called the apoenzyme, and after combining with it is called holoenzyme, which is the active form of the enzyme. So, for this reaction, the conversion of acetyl CoA to malonyl CoA, you have to identify which is the correct cofactor for this following conversion. Okay. So, let's see the answer first, then we will discuss this conversion and the cofactor. So, the correct answer is number B, biotin. Biotin is the cofactor for the conversion of the acetyl CoA to malonyl CoA. Now, what is acetyl CoA? We all know that acetyl CoA is a very important in our biological system. So, it is basically nothing but a thioester. Okay, why it is thioester? As you all know, what is the structure of an ester? And we know the structure of a ester. If it is a acetic ester, if we say that this is a structure of a ester, there is a C double bond O and O, and there is other part. So it is called ester. But here you can see there is a sulfur is present here, and it is called that's why it is called a thioester. It is a acetyl CoA. So there is a coenzyme A. There is a thiol part of the coenzyme A, and after the reaction happens with the acetic acid, so it forms the acetyl CoA, which is a very important substituent in our body. And the reaction of the conversion of acetyl CoA to malonyl CoA, it goes by this path, and there the enzyme which is the acetyl CoA carboxylase. Now, what is this acetyl CoA carboxylase does? It basically incorporates a carbon dioxide into this acetyl CoA group. So, if you see the structures of this acetyl CoA and malonyl CoA, you can see that here there is a CH3 group, and if we substitute one hydrogen with a carbon dioxide, or carboxylate group, we will have this part which is called the malonyl CoA. and this carbon dioxide, and in this reaction one ATP is converted to ADP plus Pi, and this biotin helps it helps in this incorporation of this carboxylate group into this acetyl CoA, in which reaction we get this malonyl CoA. Okay, so this is a very important reaction, and you don't need to remember the exact mechanism, but please remember the name of the enzyme acetyl CoA carboxylase, and there is one molecule of ATP which is converted into ADP plus Pi here, and biotin is the cofactor which is helping in this reaction. Okay, so I think it is clear why what is the biotin and why, and I will say just a second. Okay. Okay. So if you have any doubt or anything, please write in the chat box. And if you there is a if there is any problem in seeing my hearing my voice or seeing the presentation, please also do write in the chat box. Okay. So I think it is clear. This part, the cofactor and what is the cofactor conversion of acetyl CoA in malonyl CoA. Then we will move toward our next problem. Okay, so the problem number 3, it says that linking of the building blocks in a polyketide biosynthesis pathway is similar to a reaction of option A, Claisen condensation, option B, aldol condensation, option C, Diels-Alder reaction, and option D, Michael addition. So basically, these four are very well-known organic reactions, and in our body, the biosynthetic pathways which are happening, actually there basically there are many kind of this kind of reaction is happening in our bodies also. Okay, so among these four reaction, you have to choose the correct reaction, which is basically the similar to the polyketide biosynthetic pathway in our body. The linking of the building blocks in the polyketide biosynthetic pathway. So let's see the first what is the correct answer, and then we will discuss that reaction and this polyketide biosynthetic pathway in a detailed manner. So the correct answer is the Claisen condensation reaction. So what is a Claisen condensation reaction? The mechanism. So basically, it is a reaction in which from an alpha s from a ester from an alpha this kind of ester which has an alpha proton, we will get a beta keto ester. Okay. So what is the reaction? At first, there is a ester like this. So it should have a alpha hydrogen. So what is a alpha hydrogen here? So basically, actually, the carbon next to the carbonyl group of the ester. So this is a ester. I have discussed if there is a OET. It is a ethyl acetate kind of molecule. So if there is a carbon here next to this carbonyl group, it is called the alpha carbon. And this alpha carbon has the hydrogens which are actually very acidic in nature, which are called the very acidic alpha hydrogen. So the first step of the Claisen condensation is the deprotonation of the alpha carbon. So for the deprotonation, milder bases like this sodium NaOH, it is used, and then what happens after the deprotonation? We have a negative charge here, which is present here at the alpha position, negative N. Now what will happen? This negative charge at this alpha position is very stable, because it will take part in the resonance like this. Okay, so it is very stable, and that's why the alpha hydrogen actually are very acidic, because the conjugate base this one is very stable. Now this negative charge, it will take part in the nucleophilic attack. This will act as a nucleophile, and it will attack the electrophilic carbon of the carbonyl group. And we all know that what is a nucleophile, which is a electron rich moiety, which is called a nucleophile, and an electron deficient moiety, which is called an electrophile. So the carbonyl carbon of the ester group, it is basically electron deficient, because there if actually it is okay, somebody wants to join. Okay, so where so the carbonyl carbon of the of this ester group, it is basically very electron deficient. And why it is electron deficient? Because if we see that there is a So if there is a carbonyl carbon here, like this is a carbonyl group, and we all know that if this is a delta negative end, means because the oxygen has higher electronegativity than the carbon, so it will stay as this delta negative and delta positive. So what is the delta negative delta positive means that this electron cloud of this bond, it will be shifted toward the oxygen most of the time, which makes this carbon electron deficient, or it acts as a electrophile. Now what happens after the deprotonation, we have this negative charge which will act as a nucleophile, and it will attack this electron deficient electrophilic carbonyl carbon, and we will have this kind of resonance here. Okay. So after this happens, so what will happen now? What will happen? This negative charge comes back here to form the carbonyl carbon, and this OET group will be act as a leaving group, and it will removed from the molecule, and we will have this kind of molecule which is called a beta keto ester. Now why beta keto? I said that the carbonyl carbon of the ester, the next carbon is called the alpha carbon, and again the next carbon is called the beta carbon, and in this beta carbon we have a ketone group, so it is called a beta keto ester. Okay, so this is mainly called the Claisen condensation reaction. Now this beta keto ester has a very acidic proton. At this position we also have another acidic proton, and which is removed by the base, which can be removed. That is why for this reaction acidic workup is needed, not the basic workup. Okay. Now, so the question was, the linking of the building blocks of a polyketide biosynthetic pathway is similar to this Claisen condensation, and from this mechanism you can easily understand why I had said so. So as you can see that the first step for this, there is a acetyl coenzyme A, which we discussed in the first point, and there is a carboxylation reaction which is catalyzed by this acetyl CoA carboxylase and biotin, and we have a malonyl CoA from this acetyl CoA. Okay, now what will happen? This two malonyl CoA unit, there will be a negative there will be a negative charge as the alpha carbon after the deprotonation, and which will attack to the another carbonyl carbon of a malonyl CoA enzyme. And what will happen? We will have this kind of beta keto ester, which is, so the mechanism is similar to this Claisen condensation reaction. And like this, there will be the formation linking of the building blocks. So and we will have a chain of this polyketide, which is the polyketide biosynthetic pathway. So I think the mechanism of the Claisen condensation is clear. It is basically very easy mechanism, and this is how the reaction. You don't need to remember the exact mechanism, but it will be better if you can remember the mechanism of the Claisen condensation, because other things are very same. There is only the difference is the molecules, otherwise the mechanism is almost same as the Claisen condensation reaction. Okay, so I think the mechanism of Claisen condensation is clear. Then we will move toward our next problem. Again, I am saying, if you have any doubt regarding any part of today's lecture, please write in the chat box at the end of the session. If I get time, I will try to answer your queries, and if we don't have the time, I will share my email ID. You can mail me about your queries. Okay. So now I will move toward our next problem. So the next problem says that the sequence of the enzymes involved in the fatty acid biosynthesis is option A, ketosynthetase, ketoreductase, dehydratase, and enol reductase. Option B, dehydratase, ketoreductase, oxidoreductase, and ketosynthesis. Option C, enolyl reductase, dehydratase, ketosynthetase, and ketoreductase. And option D, ketoreductase, ketosynthetase, enonyl reductase, and dehydratase. So there are four kind of enzyme to join. There are four enzymes, and we have to just choose the correct. Okay. So there are four kind of enzymes, and we have to identify the correct sequence of these enzymes which are involved in the fatty acid biosynthesis. Okay. So let's see the answer first, then we will discuss this fatty acid biosynthesis pathway. So the correct answer means the correct sequence for the enzymes involved in the fatty acid biosynthesis is option A, ketosynthetase, ketoreductase, dehydratase, and enonyl reductase. So let's see the functions of the fatty acids. So fatty acids have four major physiological roles. Option A, fatty acids Fatty acids are building blocks of the phospholipids and glycolipids. Number two, many proteins are modified by the covalent attachment of fatty acid which target them to membrane locations. Fatty acids are fuel molecules and they are stored as triglycerols, triacyclic glycerols, and fatty acid mobilized from triglycerols are oxidized to meet the energy needs of the cell and organism. Fatty acid derivative serves as hormone and the intracellular messengers, that is steroids, sex hormones, and the prostaglandins. Okay, so these are the some important functions of fatty acids. Now we will see the fatty acid biosynthesis pathway. So what happens that there is a primer which is the acetyl ACP and malonyl ACP, which is a chain extender. Now the first step here comes the beta ketoacetyl ACP synthesis, which is in short we can say it is as a ketosynthesis. So what it happens that their first reaction happens between this malonyl ACP and acetyl ACP, and we get this 3-oxobutanoate from this reaction, and there is a elimination of carbon dioxide and ACP thiol from the first step, and we get the 3-oxobutanol. And from this, for the second step, we have this beta ketoacetyl ACP reductase, which basically converts this 3-oxobutanoate to 3-hydroxybutanoate. Means this carbonyl group which is present here, it is reduced to this CHOH group. That is the alcoholic group. And for this we have this enzyme ketoreductase have involved in the second step. And in this step, one NADPH molecule is converted to the NADP plus, and the hydro, because there it is a helps in the transfer of this hydride molecules to this ketonic group. So we have this hydroxyl group from this ketone. For the third step, there is a beta hydroxyacetyl ACP dehydratase enzyme which converts this 3-hydroxybutanoate to trans to butanoate. And in this trans to butanoate, there is a this double bond has a trans configuration, and in the last step there is a aniline ACP reductase which converts this trans to butanoate to butanoate, and in this step also there is also NADPH converted to NADP plus. And there is also a last step which is the last step for fatty acid biosynthesis, and here there will be 6 molecule of malonyl ACP converted, and we will have this type 1 fatty acid synthesis. Okay. So this was the biosynthetic pathway for the fatty acid biosynthesis, and you don't need to remember exactly each and every steps, but remembering the actually the enzymes are very important, because you can be asked to, and if you can remember this pathway, this mechanism, you don't need to remember the mechanism, but the structure of this 3-oxobutanoate, hydroxybutanoate, transbutanoate, and butanoate, it will be helpful to you. Okay. So I think it is clear the problem number 4, and now we will move toward our next problem. So the problem number 5, it says that what are the building blocks used in the biosynthesis of 6-deoxyerythronoloid B? So option A, methyl malonyl S-CoA and acetyl S-CoA, and there is option B is propenyl S-CoA and acetyl S-CoA. Option C, malonyl S-CoA and methyl mannoline S-CoA, and option D, propenyl S-CoA and methyl mannoline S-CoA. So we have to identify the correct building set of building blocks used in the biosynthesis of 6-deoxyerythronoloid B. Okay. So let's see the first answer first. Correct. So the correct answer for this problem is option D, propenyl S-CoA and methyl mannoline S-CoA. So these are the two building blocks for the biosynthesis of the 6-deoxyerythronoloid B. So here you can see it is a structure of the methyl malonyl S-CoA. As acetyl CoA, it is also a thioester. There is a it is a actually basically there is a coenzyme A, and there is a methyl manonyl acid, and we have this methyl manonyl S-CoA, which is thioester of this methyl manonyl acid. So for the methyl, actually it is a for this reaction that there is a methyl manonyl acid, and we have this propionyl CoA, acetyl propionyl CoA, and which is a thioester of the propionyl acid, and there is the enzyme deoxyerythronoloase B synthetase 1, 2, and 3, and we have this 6-deoxy erythrolyte B from this reaction. So this enzymes which basically catalyzes the reaction between these two building blocks, and we have this 6-deoxyribonucleic B, and this after is in various steps we have this erythromycin A, which is basically antibiotic, kind of antibiotic, which we discussed in our previous week. So this is this 6-deoxyribonucleic B is basically the starting material, we can say, from which we can have this erythromycin A, and this methyl mannoline CoA, and this propionyl CoA, which are these two thioesters are the building blocks for this starting material. And please remember the name of the enzyme, which is very important, deoxyribonucleic B synthesis 1, 2, 3, because you can be asked what is the enzyme which is involved in the biosynthesis of the 6-deoxyribonucleic B. So remembering the name of the enzymes are very important in your course, because the enzymes are very important because they are basically catalyzing the everything in our, what is happening in our body. So remembering the name is very important here. And this is the structures. If you can remember the structures of this starting materials, which is very good. So basically these are very easy structure. As you can see, there is only the thioester of this CoA, and the acids which are the methyl manonyl and the propionyl acids. And if you identify the names, you can easily draw the structures from the names itself. Okay. So I think it is clear. If it is, there is no problem, I will move toward our next problem. So problem number 6, it says that the enzyme responsible for the termination step of the polypeptide biosynthesis is option A, ketoreductase, option B, thioesterase, option C, oxidoreductase, and option D, dehydrase. So in the last question, I think in the problem number 4, we discussed the polypeptide biosynthesis pathway, and I said the name of the enzyme itself in that slide at the end. And if you can remember, it is a very easy question. So basically, let's see the answer. So the correct answer is B, thioesterase. As I will just move toward our last. So here it is. Now we said that this is okay. Was I? Huh. So the thioesterase, it is a very important enzyme for our polypeptide biosynthesis. So there are various type of thioesterase present in our body. The acetyl CoA is a thioesterase itself, and there is acetyl ACP, there is a glutathione and proteins also, and these are involved in various parts in our body in various mechanisms. Okay, so this is basically the reaction catalyzed by the thioesterase. So what is happens that it will just eliminate basically thioesterase. Basically, what it happens that it hydrolyzes this thioester. Like we have this is thioester part here, and what the thioesters will do, it will just by the use of a water molecule, this coenzyme A, it will be eliminated for the thioester, and we will have this acetate part only. Okay, so here is a detailed mechanism of the reactions involved in the biosynthesis and how the thioester is working. So as you can see, there is a water molecule, and after deprotonation it will have form a hydroxyl ion which will attack this carbonyl carbon, and we will have this elimination of the coenzyme A. Okay, so thioesterases are a very important class of enzymes which basically helps in our various catalyzes various reactions of our body, and this enzymes is responsible for the termination step of the polypeptide biosynthesis. Okay. So now I think it is clear that thioesterase is the enzyme which is important for the polypeptide biosynthesis. If you don't remember the exact biosynthetic pathway, it is also alright, but try to remember the key enzymes which are involved in the first step and the last steps of the reactions. Okay, because you can be asked to write the names of the enzyme, or particularly you can be given the enzyme thioesterase, and you can be asked what is the role of this enzyme of thioesterase. So these kinds of question can be asked to you from this part of the course. So I will suggest so remember the name of the enzyme and what is it does basically. So we will now move to our next problem number 7. So problem number 7, it says that which of the following statements is false regarding the virus. Option A, viruses are submicroscopic organisms. Option B, viruses only grow and reproduce inside the host cell. Option C, COVID-19 is a viral disease. And option D, viral infection is confined only a particular part of our body. Now we all know viruses we all hear about viruses now and then, and after the COVID-19 also, we are very particular about the viruses and we know much better. And so you have to identify the false statements regarding the virus. So let us see the correct answer, and means which is the false statement regarding the virus among these four, and then we will discuss about viruses. Okay. So the correct answer is the number D. Viral infection is confined only to a particular part of the body. So it is the false statements regarding the viruses among these four. So what are the virus? The definition says that a pathogen or disease causing agent not considered living because it cannot reproduce on its own. Okay, so viruses, they cannot on its own. They need the host cells to for the reproduction of the, because they can only grow and reproduce inside the host cell. Okay, so without the host cell they cannot grow, or they can go the reproduction. These are the pathogen or disease causing agent which are submicroscopic, and but they are not confined only a particular part of a body. So there is a very common confusion between the viruses and bacteria. What is the differences between the viruses and bacteria? So let us discuss the differences between the virus and bacteria. So viruses, they are not living enzymes, but bacteria, they are living organisms. So viruses are not living organisms, but bacteria, they are living organisms. Viruses only grow and reproduce inside the host cell they infect. When found outside the living cells, viruses are dormant, and their life therefore requires the hijacking of the biochemical activities of the living cell. And but bacteria are living organisms that consist of a single cell that can generate energy, make its own food, move and reproduce, typically by binary fission. This allows bacteria to live in many places like soil, water, plants, and the human body, and they serve many purposes. So we all know that there are good bacteria also, because in our body bacteria catalyze many reactions. So viruses are submicroscopic, bacteria are giant compared to the viruses. A viral infection is systematic. Viruses infect a host cell, then multiply by the thousands, leaving the host cell infecting other cells of the body. Bacteria infection are usually confined to a part of body, described as a localized infection. Infection may be caused by the bacteria or by the toxin it is producing. So it is a very important point that virus infection is systematic. The virus, they will infect the host step, then will multiply and reinfecting other cell in the body. So if we have a viral infection in a particular part of body, it will just infect other cells of the bodies too. But the bacterial infection, it usually confined to a part of a body. That's why it is called the localized infection. So this is a very key crucial difference between the viruses and bacteria, which you need to remember. The systematic diseases caused by viral infection involve the influenza, measles, polio, AIDS, and COVID-19. And for the bacterial the diseases, they include pneumonia, tuberculosis, tetanus, and food poisoning. So there is also another reason why we cannot have antivirus, antibacteria kind of, because virus use body's own mechanism. So it is a very difficult to actually form a drug molecule for the viruses, but for the bacteria it is actually easier more than the virus. So please remember these differences between the virus and bacteria, especially number two, which is the that viruses only grow and reproduce inside the host cell, but outside the host, it does not have any power to grow or reproduce. But the bacteria can live of its own, and that's why we have in soil, water, plants. We have in human body we have bacteria every place. And the viral infection, it is systematic, but the bacterial infection, it is localized. Okay. So I think it is clear the differences between virus and bacteria. Then we will move to our next problem. So problem number 8, it says that Baltimore, Nobel Prize winning biologist, classified viruses in dash groups. So you have to identify how many groups this Nobel Prize winning biologist has divided this viruses. Okay. So the options are number A 4, option B 6, option C 7, and option D 8. So you have to identify the number of classes in which the viruses are divided by this biologist. Let us see the answer, then we will discuss this groups and about this. So the correct answer is number C 7. So this Nobel Prize winning biologist has classified viruses into 7 groups. So let us see what are the 7 groups are present. Okay. So there can be 3 kind of viruses. One can be DNA viruses, okay, and there is a RNA viruses and reverse transcripting viruses. So for the group one, we have double-stranded DNA. And for the group two, we have single-strand DNA, and or double-strand from this group to the single-strand DNA can be double-strand DNA, and this DNA is actually infect this in our infect this body, and then this replicate. And for the RNA viruses, we have the double-stranded RNA, and which is the group 3. We have single-strand RNA, which is group 4, and we have single-stranded RNA, which can be positive or negative. So it has a group 4 and group 5. This positive and negative is divided. And for the single-stranded, we only have this positive, which is the group 2. And for the there is reverse transcripting viruses, and there is a single-stranded RNA and the double-stranded DNA. So which are the group 6 and group 7. So these are the 7 groups, and I will just say that please remember that there are 3 kind of viruses. One is DNA viruses, RNA viruses, and reverse transcripting viruses. And this classic groups, so if you can remember these groups, it will be very helpful because it is a very important part of the group. So please remember that this there are 7 groups, and which group has which kind of DNA. Like group 1 has double-stranded DNA, and group 2 has single-strand DNA positive, and the group 3 has a double strand RNA positive or negative, and like this remember each and every group, and also this 3 classifications. Okay, so I will now move toward our next problem. So if you have any doubt, please write in the chat box. At the end of the session, we will discuss those doubts, and also I will discuss. I will just share with my email ID if you have any doubt regarding this class after the end of the session, and we can discuss in the email also. So the problem number 9, it says that which of the following statements is false regarding the retrovirus? Option A, it contains RNA as genetic material. Option B, reverse transcriptase enzyme is present. Option C, it proceeds through the DNA independent steps. And option D is none of the above. So you have to know what is the correct false statement regarding the retroviruses. The retroviruses are a very important class of important class of the viruses, and you have to identify the correct one for the correct statement about the retrovirus. Okay. So the options correct answer is the option C. It proceeds to the DNA independent steps. Okay, so there are some interesting points of the retroviruses. So number A is the retroviruses contain RNA as generic material but have DNA independent step in the replications. These replicates via reverse transcription because of the presence of the reverse transcriptase enzyme. The integrase transfers the viral DNA into the cell nucleus and the viral double-stranded DNA as the covalently and randomly integrated into cell's genome. The retroviruses that can transform host cells at high rates contain gene sequences such as viral oncogenes and the proto-oncogenes. And human retroviruses can cause immune deficiencies, cancer, and neurological diseases. Okay. So these are the interesting points of the retroviruses, and perhaps the first one is the most important, that the retroviruses, they contain RNA as a genetic material but have DNA independent step in their replication, and they just replicates via the reverse transcription which in presence of the reverse transcriptase enzyme. So this is the basic structure of this retroviruses, and there is, as you can see, there is an envelope glycoprotein, there is a lipid envelope, then there is a protease enzyme, RNA matrix, there is a code, and reverse transcriptase enzymes are present here, and there is the integrase and nucleoplast. So protease, reverse transcriptase, integrase, these are a few important enzymes which are involved in this retroviruses. Okay, and inhibiting this kind of enzymes also are a part for the drug development mechanism. So now I think it is clear that which one the statements is false regarding the retrovirus. Please remember these points regarding the retrovirus and the roles of the enzyme, like the integrase transfers the viral DNA into the cell nucleus, and the viral double-strand DNA is covalently and randomly integrated into the cell's genome. Okay. So if there is no problem, I will just move toward our next problem. So problem number 10, it says that acyclovir, the structure shown below, which is this is the structure of the acyclovir. Okay, this is the structure of the acyclovir, is an inhibitor of DNA polymerase selectively in virus infected cell. So this is because option A, acyclovir is first converted to monophosphate only by the viral thymidine kinase. Option B, acyclovir does not enter infected cells. Option C, acyclovir is first converted to monophosphate by only the host thymidine kinase. And option D, acyclovir triphosphate is not accepted by the host DNA polymerase. So you have to identify what is the correct answer and why acyclovir is an inhibitor of DNA polymerase selectively in the virus infected cells. And this is the structure of the acyclovir, which is a drug molecule. Now let's see the correct answer. So the correct answer is number A. Acyclovir is first converted to monophosphate by the viral thymidine kinase. So actually, this acyclovir, it is called a prodrug because it is not an actual drug. It is first converted to the monophosphate only by the viral thymidine kinase, and this prodrug will be converted to the drug molecule by the viral thymidine kinase, and by this mechanism this acyclovir inhibits the DNA polymerase in the viral infected cell. So it is very important that we also, this human body also have this DNA polymerase enzyme, as we all know. So because for a drug molecule, for inhibitor for this DNA polymerase selectively only in virus cells is very important. So why that there is a acyclovir works by here, because it is only converted into the monophosphate by the viral thymidine enzyme kinase. So in our body it is given as a prodrug. So as I said that this acyclovir, it is a prodrug. It is not a drug molecule, actually it is called a prodrug molecule, and it is converted into the active form of the drug by the only the viral thymidine kinase enzyme. Okay, so this enzyme is not present in the our human body, and this acyclovir, it is not converted into this active form by the human kinase. That's why it cannot inhibit the human DNA polymerase enzyme, but selectively it inhibits the viral DNA polymerase enzyme. That is because this viral thymidine kinase, it converts this prodrug acyclovir to its active form to the monophosphate. Okay. So this acyclovir drug, it is a very important drug, and please remember how it works, and this is a very important question because of that. And the structure of acyclovir, if you can remember the structure, it is very good because it is a very important topic. Okay, so I think it is clear how the acyclovir work and why it is only inhibiting the virus infected cells, because it selectively inhibits the DNA polymerase of the virus, not the human body. Actually, this is kind of a enzymes, and for the drugs we are using for this viral molecules in viral infections, it is very important that they do not hamper the human body also, because the viruses, the life cycle of the viruses in the human body, it is very similar to the human body itself, because so the enzymes are more or less same. So designing a drug is very difficult, so that we have to design a drug by which the human body enzymes are not hampered, but only the selectively viral infected cells are hampered. Okay, so that's why it is a very difficult task to do. So if there is no problem, I will more quickly move toward our next problem. So problem number 11, it says that which of the following is not an anti-HIV drug? So option A, nevirapine. Option B, ridenosine. Option C, dihydroartemisinin, and option D, zidovudine. So you have to identify among these four which is not an anti-HIV drug. So HIV, we all know, the human immunodeficiency virus, which is a very important or well-known virus disease, and the enzyme which is the HIV, the AIDS, this HIV is a reverse transcriptase virus, and you have to identify the correct drug which is not a anti-HIV drug among these four. So the correct answer is the option C, dehydroartemisinin. So let us see the structures of the drugs first. So these are the structures for this drugs. So these three drugs, nevirapine, didenosine, and the zerovodin, these three are the anti-HIV drugs. Okay, so they inhibits this human immunodeficiency virus, and by that there are various mechanisms. There are different mechanisms for the and but this number three means the dehydroartemisinin. It is a anti-malarial drug. As you, I don't know if you know or not, but the artemisinin is actually is a traditional Chinese medicine, which is nowadays recommended by WHO, and this is called the ACT or artemisinin combination therapy for the treatment of the malaria. Okay, and dehydroartemisinin is basically a derivative of the parent drug artemisinin, and there is artemisinin, artesunate, and all their drugs. So these drugs are used for the malaria only. Okay, these are not the anti-HIV drug. So there are these three drugs: nevirapine, didenosine, and this zerovodin. These are the anti-HIV drug, but dehydroartemisinin is the anti-malarial drug. So I know there are many names of the drugs where it is sometimes it is difficult, but I will suggest please remember few names of the drug. Like if you are given that nevirapine, which kind of drug it is, and you should know the answer that it is a anti-HIV drug. Especially this nevirapine is a very important anti-HIV drug, and artemisinin is a very important anti-malarial drug. So the question can be given you by directly by the name of the drugs, like what is the role of dehydroartemisinin? What is the is it an antibacterial, anti-malarial, or antiviral? So you should know that it is an anti-malarial drug, and the nevirapine and this of these two drugs, these are the anti-HIV drugs. Okay. And remembering the structures is not that important, that much important, since in this course you don't have a very detailed synthesis, but if you can remember one or two structures of the drugs, it is very good, especially if you can remember the structure of nevirapine, and I will suggest you to remember only the one or two structures, it will be enough, okay, for this course. So now I will move toward our next problem. So problem number 12, it says that which of the following statements is true regarding the HIV protease enzyme? Option A, it belongs to the enzyme family of the cysteine protease. Option B, it catalyzes the formation of the peptide bonds. Option C, active site structure contains cysteine moiety. And option D, active site structure contains the aspartic acid. So HIV protease enzyme, it is a very important enzyme for this HIV virus, which is basically causes the AIDS. And there are, I said in the previous slide, one of the slide, that for this retrovirus, it is a retrovirus. There are several enzymes which there is a DNA polymerase, protease, there is an integrase. So this kind of enzymes, they are very important for the survival of the HIV virus, and by inhibiting this kind of enzymes, by designing the inhibitor of these enzymes, we can actually design a drug for the HIV AIDS. Okay, and this is the current line of the research what is going on for the HIV AIDS. So HIV protease is a very important enzyme for the survival, and you have to identify the correct statement regarding this HIV protease enzyme. So let us see the correct answer first, and then we will discuss the problem. So the correct answer is option D. The active site structure contains aspartic acid. So for the HIV protease enzyme, in the active site structure it contains the acetic acid. So as I said said before that, okay, so as before I have said that this HIV proteases are very important enzyme for the survival of this human immune deficiency virus. So as you can see in this picture, that there is a one long chain of HIV protein inside the infected cell, and what does this protease does? They basically cuts this HIV protein into the smaller parts. Okay, and the this individual parts, they can they enzymes that individual proteins, they are make new HIV particles, and individual enzymes that help to build this new HIV particles. by inhibiting this protease enzyme, we can actually we can stop this formation of individual smaller chains of protein of new HIV particles. So basically, protease inhibitor drugs, they basically inhibits this protease enzyme. And as you can see in this structure, that there is a aspartic acid residue which are present in the active side of the protease HIV protease. And these are the remember the few drug molecules for which are the HIV protease inhibitors: the indinavir, nelfinavir, ritonavir, sacuavir, and emrivir. So the remembering the names of the drugs are actually very interesting. That you can see that if there is a vir at the end, okay, in each of the drug they have this this navir kind of this sequence in the at the end. So if you are given a name for, like if you are giving the name Indiavir, if you just remember that these are example of HIV protease inhibitor, so it will be very easy. And remembering the structure of the drug molecule is not necessary, because it is not possible to remember each and every structure, and these are huge structures for these drug molecules. But the novices, please remember the examples of the drugs for the HIV protease inhibitors, because in the exam you can be asked to give the example of few HIV protease inhibitor drugs, and you also can be given the names, and can be asked that what kind of drug molecule it is and what kind of antiviral molecule it is. Okay. So, HIV protein is a very important class of the enzyme. Remember how it works, and the active site, it contains the aspartic acid. You don't need to remember the exact active site structure, and also please remember the few important examples of this kind of HIV protease inhibitors. Okay. So I think I am clear what is the HIV protease enzyme, and still if you have any doubt, please write in the chat box at the end of the session. We will discuss those doubts. Okay, so problem number 13, it says that nevirapine is a drug which is an dash inhibitor of the reverse transcriptase. And the options are: option A, allosteric; option B, non-nucleoside; option C, non-competitive; and option D, all of these. So in our previous slides, we were discussing about nevirapine, and I showed you the structure of nevirapine and said that it is a very important drug, because it is a inhibitor of this reverse transcriptase enzyme. And reverse transcriptase enzyme actually basically very important part for this retroviruses. And let's see the correct answer. So the correct answer is D, all of this. So this is the structure of the nevirapine. And okay. So basically this nevirapine, it is called the non-nucleoside reverse transcriptase enzyme. Okay. So what is the mechanism of action? They act by stopping the HIV production by binding directly to the reverse transcriptase and preventing the conversion of the RNA to DNA. So why we know that the reverse transcriptase enzyme, it is catalyzed to the formation of the RNA to DNA in the virus cells, and this nevirapine is a direct inhibitor of this reverse transcriptase enzyme. They act by stopping this HIV production by binding directly to the reverse transcriptase, and they do not require the activation through the phosphorylation. So this is called there is a allosteric inhibitor. Now what is the allosteric inhibitor? So we all know that if it is an enzyme where the substrate bind, it is called an active site, as you can see in this structure. Okay, I will just, okay. So it is the active site of the enzyme where the substrate binds. Okay. So at the there is a allosteric site also, then which is not an active site, but if there is a inhibitor site present, it will if we use for what is the allosteric inhibitor, it is a molecule which binds in the allosteric site, which is not an active site, but after binding with this allosteric inhibitor, the active site's shape changes. And as the active site shape changes, the substrate will not bind to the active site anymore. Okay, so it is an allosteric inhibitor. It is not directly actually inhibiting the active site, not directly binding to the active site, but it is binding to the allosteric site. And by binding to the allosteric site, what happens? The active site structure changes, and the substrate now cannot bind to the active site. Now, what is a non-competitive inhibitor? In the non-competitive inhibition, inhibitor binds on the site of the active site. It could be the allosteric site. Okay. The non-competitive is distinguished from the general mixed inhibition is that the inhibitor has equal affinity for the enzyme and enzyme stuff. So these are the kinetics and Michaelis-Menten constant. These are the Michaelis-Menten graphs for this non-competitive inhibitor. So basically, allosteric inhibitor, they can also be called the non-competitive inhibitor. Okay, so actually, if it is bind to the another part, not the allosteric side, it is called the non-competitive, and on nucleoside also it is a part of a allosteric or non-competitive inhibitor. So this nevirapine, it actually does not binds exactly to the active sites for this reverse transcriptase enzyme, but it binds to allosteric site, a site which is not active site, and that's how it changes the active site structure for this reverse transcriptase, and by this it can inhibit the function of this reverse transcriptase. And this is a very important drug. I have said you before that nevirapine is a very important class of anti-HIV drug. So I think it is clear what is nevirapine and how it works. Now we will quickly move to our next problem. So the problem number 14, it says that the only DNA antisense molecule that has been approved as antiviral drug, it is what? A. Nevirapine, option B. Formiversen, option C. Darunavir, and option D. Zerobody. So there is a only DNA antisense molecule which has been approved to the as a antiviral drug, and the correct answer is option B, Formiversen. So it is the only DNA antisense molecule that has been approved as the antiviral drug. So what is the antisense oligonucleotide therapy? So basically there is a DNA, the viral DNA, which by the transcription can be formation from the mRNA, and by the translation this mRNA forms a disease associated protein. Okay, so what this DNA antisense molecule, what they drug actually, these are the oligonucleotides, and they will the RNAs degrades the RNA and not transcriptase. So this antisense drug, they what will they do? They will just degrade this RNA molecule, and by the degrading this RNA molecule, they will there no disease associated proteins will be produced now. So from the DNA, the RNA is formed, but this DNA antisense molecule, which will attach to the RNA, and the RNA is degrades, and no translation takes part, and since there is no translation, no disease associated protein will be formed. Okay, so this is the formiversin is the only example for the DNA antisense molecule that has been approved by antiviral drug till now. So it is very important example. You need to remember this is the structure for this formiversin, though it is not possible to remember the structure, but just know what is the antisense oligonucleotide therapy. So this is the antisense drug, it is oligonucleotide, and it binds to the mRNA produced from the viral DNA, and this degrades by the formation of this antisense drug. It binds to the RNA and degrades the RNA so that no disease associated protein is produced. Okay, so remember the name of the formiversin. Well, it is very important because it is the only DNA antisense molecule that has been approved as an antiviral drug, and how it is working. Basically, no disease associated protein is produced because of this drug. So now we will move toward the last problem of today's session. So the problem number 15, it says that which of the following statements is false regarding darunavir. So option A, it is a HIV protease inhibitor. Option B, it binds with aspartate 25 and 25 dash while interacting with the HIV protease. And option C, it has R configuration. And option D, it is synthesized by Professor Arun Ghosh. So you have to identify the false statement regarding the darunavir. It is also anti-HIV drug. As I have mentioned that if there is a suffix like this vir, then it is the anti-HIV drug. I need to remember. I just gave you the example in the previous slides. There is a easier way to remember this. So quickly we will see the correct answer. So the correct answer, rather the false statement regarding the darunavir is it has R configuration. So it is the incorrect option. I mean, it is a false statement regarding darunavir. So this is the structure of darunavir, and actually this is called darunavir because the Professor Dr. Arun Ghosh, he has actually synthesized this molecule, and from from his name, the naming nomenclature of this molecule has been done. and it has actually it does not have a R configuration, it has a S configuration, and this is the pathway by this darunavir molecule was synthesized by Professor Ghosh, and you don't need to remember the exact pathway of the mechanism, but remember that it has a R configuration and it is a HIV protease inhibitor. It is very important. Like I said you that if it has this kind of vir prefix suffix, it will be HIV protease inhibitor. So it is very important to remember that it is a HIV protease inhibitor. And if you can remember the structure of darunavir, it will be it is very good. If you cannot, it is also okay. If you do not remember the name, remember the structure. Okay. So as you can see that there is a this is a there is a nitrogen molecule. So if you have this naming of the like how we do the RS configuration, that this will be given one this nitrogen because nitrogen is higher priority than the carbon. Now this part will be given the number 2 because it has the oxygen molecule at the carbon, and this will be given 3. So if we go from 1, 2 and 3, we have a anti-clockwise motion, so it has a S configuration. Okay. Similarly, you can also see from this part also that actually from this carbon it has a S configuration and it does not have a R configuration. So remember it is about the darunavir. It is a HIV protease inhibitor, and this is the by this method it was synthesized by Professor Arun Ghosh, and so you don't need to remember the exact pathway, but I just has shown you for your just for the knowing. So that this is it. This was all about today's lecture. So if you have any doubt regarding today's class, please write in the chat box at the end of in the chat box, or I will just share my email ID with you. Okay. So in the chat box, I have shared my email ID. So if you have any doubt regarding today's session at any problem, so feel free to contact me by the mail. I will answer your query. And if you have again, if you have any problem, uh, okay, somebody has. Okay, please can you just write in the chat box if you what you have doubt, because it will be then easier for me. Please write in the chat box what is your doubt. Anyways, if you have queries after the class also, you can just drop a mail to me, or you can write your query in the grievance portal in the NPTEL website. Okay. So if you have any doubt, please write in the chat box. This was the week 11 problem solving session. I will share today's class video and this PPT in your this PPT and this class video in the NPTEL website. It will be shared to you in the due time. And if you have any doubt, feel free to contact me. So next Friday we will have our last session of this NPTEL last live session for the NPTEL course of Organic Chemistry and Biology and Drug Development. And so for now, I will just close today's session. So thank you all for joining today's class. We will meet in the next week at the same time from the Friday at 7 to 8 p.m. Okay. So thank you all for joining today's class. Now I will close today's session.